Yeah, hello, welcome everyone to our today's Amplifying Voices session. I'm very happy to introduce to you two young researchers from India. They are both working at the Institute. I'm sorry, here at the Institute of Science and Education and Research at Kolkata in the Integrative Taxonomy and Microbial Ecology Research Group in the Department of Biological Science. And we want to start with Arkaprava Mandal. He graduated in 2015 from the University Netaji Maha. Oh boy, I'm really sorry. I'm not really able to.
Okay.
From West Bengal in India, right? Maybe
Yeah.
you can tell us a bit more about it. And in zoology, and also he did his master's in zoology and is now working on his PhD on integrative taxonomy and microbial ecology in this research group that I already mentioned. And yeah, we're very, very much looking forward to hear what you're actually working about on. The floor is yours. Thank you.
Thank you. Thank you. Nice to meet all of you. Today I want to share my experience of my research work with all of you. With everyone. Okay, now I am sharing my screen. Yeah, is it clearly visible?
Yeah, it's perfect.
Okay. I am Arkaprava. I'm currently working in ISR Kolkata. It's a renowned institute in India. I am now currently pursuing my doctoral research in ISR Kolkata. I am a benthic ecologist, and my work mainly focus on the benthic ecology and its application in climate shift, climate change, sea level rise. Mainly, I am working on how climate change affect the coastal Bay of Bengal of northern Indian Ocean part. It is my primary work. Also, I am looking down the sea level rise and how the sea level rise affect on the coastal ecosystem of Bay of Bengal. So today I am share some of my work with all of you. So here is the title of my work, is that tracking the ecological health of coastal ecosystem from the northeast Bay of Bengal and its applicability of benthic foraminifera as a bioproxy. As I said, I am a benthic ecologist, so I choose a benthic organism, that is foraminifera, to track down the ecological health of the coastal ecosystem of Northeast Bay of Bengal. Yes. Well, all of we know that the United Nations set up goals to prosper of the life of our Earth. My work also fulfills the two of the goals, like the climate goal 13, the climate action, and the goal 14, the life below water. My entire study is only focused on how the climate shift changes or how the climate changes effect on the coastal Bay of Bengal. To track down the changes in the coastal Bay of Bengal, I choose an organism that is known as foraminifera. It is a single-cell eukaryotic organism, and it's a beautiful taste or cell body that covers this organism. This organism have some unique power, like that it track down any type of environmental changes that happening in the coastal ecosystem, like the erosion or the sea level rise, anything that happens in the surrounding of this organism. This organism has a power to copy that thing in their taste. That taste means the outer portion of their body. It's a single cell eukaryotic organism, and there is a hard body around this type. Then foraminifera mainly are two types that found in the marine ecosystem, also found in the freshwater ecosystem, but I choose the marine foraminifera. In foraminifera, there is two types. One is a planktonic form and the benthic form. But my study only focus on the benthic forms. The foramina first appeared in our history, the Cambrian period. After that, in the Ordovician, it diversified in two forms, the calcareous and aggregated. And in the later time, the aggregated forms and calcareous form also diversified. As you all know that every organism has a key role in the food chain. Usually the foram also play a particular key role in the food chain. It channelizes the energy from lower trophic level to higher trophic level. Foraminifera mainly feed upon on the decaying mangrove leaf, the planktonic detritus, and the suspended particulate matter. And the some of the predators that take foram as a food, like the deposit feeder like crabs, some mollusks, or some small fishes also feed upon on the foraminifera. So that's mean the foraminifera is a key player to channelize the energy system in the food web. My work encompasses a huge sampling point that covers on the seasonal basis on the northeast part of coastal Bay of Bengal, Indian Ocean. So my work mainly focus on two long-term objectives. My first objective of my work is that deciphering the ecological health of coastal Bay of Bengal by using foraminifera as a bioproxy. And my second objective, and that is the exploring the biogeocomplexity across the coastal biotas of the coastal Bay of Bengal, northern Indian Ocean. Based upon these two objectives, my entire work was done. So here I today mainly focusing on the deciphering the ecological health of coastal Bay of Bengal by using benthic foraminifera as bioproxy. As I said, my study site is the Northeast Bay of Bengal. The whole stretch of Northeast Bay of Bengal is my study site. In the stretches, I done a monthly sampling on last 2 years, and in the study site, I done transact basis sampling. That's when I choose two transact. One is the lower tidal transact that I represent as JPL, and one transact is my higher tidal area transact that lies into the coastal vegetation that I also denote as JPH. Here are the glimpse of my study site where I work. So this is the methodology that I adopt to done my work in my doctoral research. So in the field, I do some biogeochemical measurement, and I also collect samples like surface water of coastal surface water and the sediment. The surface water we have collect in further use for the dissolved nutrient estimation and dissolved organic carbon estimation, also in the rare earth metal analysis. And I also collect the sediment to track down the changes, what is changes happening in the sediment by using or by looking the foraminifera. The sediment which I have collected in the Kora, the sediment was after that treated with some chemicals like rose Bengal or molecular ethanol to preserve the foram intact in the sediment. After that, the sediment also used for the carbon analysis and the nutrient profile analysis. Here are the images, how I generally collect the sample throughout the coastal tract of Northeast Bay of Bengal and do the biogeochemical measurements. Okay, now I come to some important portion of my findings that here is showing that there's graph showing the atmospheric temperature or air temperature and surface water temperature. The air temperature and surface water temperature clearly display the monsoonal effect of my study site. In my study site, there is a very much clearly effect of monsoon. In the post-monsoon time, the air temperature and surface water temperature is low. That's usual thing. The monsoon is slightly higher in temperature than the post monsoon. The clear division or the clear effect of monsoon or the seasonal precipitation that is very much prominent in my study site, which is described by the salinity. In monsoon time, the salinity in the study site becomes 0 because the Northeast Bay of Bengal is very much influenced by freshwater inflow. That's when the Northeast Bay of Bengal receive a huge amount of fresh water from their upward area. As a result, in the monsoon time, sometimes the salinity becomes 0, and in the post monsoon time, the salinity sometimes 20 or 20 above 20 salinity. As earlier I said that I collected surface water to do nutrient profile. In my lab, I do the dissolved nitrogen, dissolved phosphorus, and dissolved silicate analysis of throughout the study area surface water. And here the result of my analysis, which was done on the seasonal basis. And this graph shows that the dissolved nutrient profile of my surface water that I have collected from the study area. Here the nitrogen phosphorus ratio is markedly above the red field ratio. That is the one of the interesting point of the study area, that the nitrogen phosphorus ratio is above the red field ratio. Also from the study side, I collected sediment to textural analysis. In the benthic ecology, the textual analysis help us to understand the changes that happening in the sediment, and that what is the happening in the sediment also affect on the pelagic ecosystem. That means that there is a connection, the benthic pelagic coupling, that all we know, because in the sediment is something wrong is happening that affect on the pelagic part. If there is any something is happening or not happening, to understand this thing, the sediment texture analysis help us a lot. So I do a lower sampling to understand the sediment texture profile of my studied area, and it shows the coastal Bay of Bengal, which in my studied area is predominantly sand. That means the studied area have the sediment composition sand dominated. After that, I do the benthic foraminifera profiling. As I earlier said that I interested in the benthic form of foraminifera, and the benthic form of foraminifera is a good indicator of sea level rise or anthropogenic stress or erosion, etc. To understand that this type of changes which is actually happening or not happening, we have to do the profiling of benthic foraminifera. So then I do a 2 years robust sampling to create a benthic foraminifera profile. And here I show some of the important study sites and the benthic profiling of the study site. Here you show some few common taxa that found in the studied area, like Astereolotalia crispinosana, ammonia, Polyphena elphidia. In my study site, this Astereolotalia crispinosana is dominant. After that, the ammonia species is second dominant. This ammonia species has a particular role. That's when the ammonia species is an indicator of stress in pollution or any type of stress like erosion or polyhydrocarbon pollution or rare earth metal pollution, like that. That's mean the ammonia is a stress loving species. That's mean if in my study area the ammonia population is predominantly higher than other species, that's mean the study area is under threat. But thank God in coastal Bay of Bengal is not as much as under threat. There are the few bright field images of benthic foraminifera that I usually encounter in my studied area. This is the Astereolotalia crispinosana, this is the Polyphena and this one is the ammonia, and this one is the quinquillina. These are the common benthic foraminifera found in my studied area. Okay, now I come to some interesting features of benthic foraminifera. That the benthic foraminifera also subdivided in two groups. One is calcareous and another is arcunated. These two name was depend on the composition of their cell structure. That means, as I earlier said, every foram has a hard cell part that protect them from any type of external danger. And this test or cell are made of some component. Based on this component, it divided into two parts: calcareous and arcunated. In any system, if the calcareous and arcunated foraminifera ratio is disbalanced, that means some of form is over dominant with other, that indicating the study area is under some type of disturbance. In my study area, one notable thing is that the calcareous form is over dominated on the arcunated form, that indicating there is something disturbance is happening in those area. Probably the fresh water or marine water mixing, or the influx of fresh water is low, or the fresh water marine water mixing is not so good. That means the marine water influence is too much higher than the fresh water. Anything possibility is that, but one thing is clear: the calcareous form is over dominated and arcunated form. That means there is something disturbance happening in the study area. I am now working how or what type of disturbance is happening in this part. Okay, the second interesting feature of this foraminifera is that it marks or it copy anything unusual that happening in the study area. That means if the sediment of the study area become anoxic or suboxic, or there is something toxic substances formed in the sediment that affect on the foraminifera test, it makes the foraminifera test degraded. That means if in the sediment the anoxic condition is there any anoxic condition, or is there any hypoxic condition, and that leads to the production of hydrogen sulfide or ammonia, and that ultimately makes the sediment toxic, that ultimately affect on the test and make it degraded. In a nutshell, if we look through the degraded test, we can interpret the ecological health of in studied area. Here the profile of dissolved nutrient test or degraded test that I encountered during my study time, and this is the graph what percentage of dissolved test was encountered in my studied area, and this is the image that degraded test, how degraded test looks alike. In my studied area, the degraded test, each number of degraded test is increasing day by day. That means the studied area under threat. Okay, then one of my part of my study is that how sedimentary organic carbon helps in foraminifera population, helps to boost up the foraminifera population, or is there any significant relationship between the total organic carbon of sediment or sedimentary organic carbon and the foraminifera? That's why I selected a dominant foram form that encountered in the study area and do some statistics to show is there any relationship between them. But my statistics said that in this area there is as such no significance role or significance importance of total organic carbon or sedimentary organic carbon on the population of asteroid area. Okay, now come to the major finding that my study area is completely dominated by sand or silt, and the benthic foraminifera diversity or abundance in my study area is low rather than the similar settings of the world. And in my study areas, the calcareous form is over-dominated on the agglutinated form, that indicating the studied ecosystem are prone to severe disturbance. And the lower abundance of agglutinated population also indicating the rapid degradation of ecosystem quality. And day by day, the presence of degraded taste is increasing, and the presence of higher number of degraded taste indicating the taphonomic alteration that's happening in the sediment. That taphonomic alteration means production of hydrogen sulfide or production of toxic ammonium that ultimately affect the pelagic ecosystem or the benthic pelagic ecosystem. And the last and least point, that sedimentary organic carbon did not play a significant role in the benthic foraminifera abundance in my studied area. I am very much grateful to my lab, Integrated Taxonomy and Microbial Research Group, to my lab colleagues helping me to do my works and field works. And I also grateful to ASLO Kolkata and the Ministry of Education, Government of India, Ministry of Environment, Forest and Climate Change, Worldwide Fund for Nature India, National Tiger Conservation Authority, and the Wildlife Trust of India. Thank you.
Thank you. Thank you very much for that very interesting insights into a system that for, I guess, most of us is very far away, both like from the distance, but also like from the insights that you gave. Thanks a lot. So are there questions?
Yeah.
There's Roxanne. Please go ahead.
Yes, hello.
Hi.
Thank you very much for this really interesting presentation. I have a question about the agglutinated forms of foraminifera, because your premise is this represents ecosystem degradation.
Yes,
Can you qualify this to me as to why you're seeing, what conditions in your system are favoring that, and as well as how does this compare to other sites, other coastal sites? Are you seeing it a lot more, or is it just kind of through certain types of seasonal effects? Is there something that you can elaborate on that for me, please?
Yes. My system is a freshwater dominated system, and like in Indonesian, the country Indonesia, or in the South Africa, where the freshwater dominated coastal ecosystem. In the freshwater dominated ecosystem, the ecosystem is more dynamic, and as a result, there is a lot more happening in the coastal part. That changes the nutrient stoichiometry or the biogeochemistry of the coastal area, and this organism which I have studied have a power to copy these changes in their taste. Obviously, there is a effect of precipitation on the foram forms and the abundance, but that's not means the seasonal precipitation is one of the cause that affects on their taste degradation or taste abnormality. Beside it, there is other factors like the heavy metals, that's mean rare earth metals like arsenic, cadmium, zinc, also affect on the dissolved taste of the polyhydrocarbon. That's if there is contamination of polyhydrocarbon in the sediment, also affect the taste of foraminifera. So that's mean the seasonal precipitation obviously affect the abundance or the profile of foram, but it is not only one factor that affect its abundance or diversity, rather than other things like sea level rise or the erosion also plays a role in the study area. That's why the foram population or the foram diversity changes.
So I just have a follow-up on that, because I think it's,
Yes,
the
yes.
monsoon is really interesting because it brings in this
Yes.
huge amount of fresh
Yes.
water together with different contaminants, right?
In the Indian Ocean part, here is the most of the fresh water deposited in this part of Indian Ocean, the Northeast Bay of Bengal. Huge amount of fresh water that was debouches in the Northeast Bay of Bengal. So in this area, the monsoon plays also a significant role.
But I guess I think it might be interesting. I don't know if anyone's doing this, but
Yes.
in temperate systems, coastal systems, you could have a huge amount of fresh water coming in after snow melt. That might have sort of
in temperate system, yes.
Yeah, but I guess it's tough to go and measure foraminifera during those moments. But anyway, but thank you very much.
Yeah, thank you. Thank you.
Are there further questions?
I had one question if there's a minute.
Yeah, of course, go ahead.
Thanks for that presentation. And I just following up on the last question about, is this the first time that these organisms have been measured in this area? Are there other baseline studies that?
No, it is not a first study. Also, researchers from our country to the theme, but my study has some uniqueness. I cover a long distance, and I am doing a huge sampling and covering a huge number of sampling points. These are the two uniqueness of my study. I am doing a long-term study that is first, and then I am covering a huge number of sampling points that covering a discrete gradients of hydrodynamics of this area.
Yeah. Okay. Thank you. Yeah, it's very impressive, the number of parameters that you're able to measure, and I'm glad that you're doing it over the long term, because I think that's an important part of biomonitoring studies. So you have to have a baseline to then detect changes. So,
yes.
Thank you very much.
Yeah. Thank you.
Any more questions there? There is one question coming in that is from Abhishek. How long has the site been studied in order to establish the relationship between trends in measurements and climate change?
On the how long the studies has to be, okay. In order
to establish the relationship between trends in measurements and climate change. Maybe the person
in my in my understanding, minimum 1 year study is important to understand or to glimpse the changes that is happening due to climate shifting or other thing. Minimum 1 year. That's my understanding. But I think that if you study the long term, that obviously good. From a short term study, there is no doubt, but minimum 1 year study is required.
Yeah, I guess that's one of the limitations that we have in PhD studies that are only financed for a few years, and that's one problem. We are running a bit off top, but there's one more question. There's a few more questions actually. Let's quickly go for them. Is there a relationship between temperature and foraminifera abundance? Is a question from Evaristo.
Yes, there is a relationship between the surface water temperature and the foraminifera abundance. Global warming that affects on the surface water temperature, and the changes in the surface water temperature also affect the foram growth, the foram reproduction. And this type of study already has been conducted in the deep sea, but this type of study is not very much popular in the intertidal zones, but it is already done in the deep sea areas.
Thank you. And then we have one more question from Dinah as a follow-up to one of the questions before, I guess. Do these organisms preserve in the sediment such that you can estimate rates of change over time at different sites?
Yes, for this type of study, we all conducted the microcosm study and in the lab to track down the changes, how the time takes to change in their uh, cells. And the microcosm study was done, and we also now try to do something of that in both in study site and in laboratory. And this type of study will basically very much helpful to understand the how times or how long it takes that the changes happen in the taste of forams.
Okay, thank you. With this, I want to thank you overall, especially to you, Arkaprava, for this great introduction to your topic and your work. And
Thank you.
yeah, you did a great job here. And then we want to switch to your colleague, Chakresh.
Yes, Chakresh.
Exactly.
Then I am stopped my sharing.
Yeah, exactly. That would be great. And Chakresh.
Yeah.
Kumar. Hello
Yes.
there again.
I am visible?
Yes, you're visible and we can hear you. That's great. You studied environmental and water management at Gaia College in India, and then went ahead with a master's in environmental science to now also work in the Department of Biological Science at the Indian Institute of Science Education and Research, Kolkata, on your project that you want to introduce with the talk title that is Seasonal Dynamics of Environmental Variables and Water Quality Index in the lower stretch of the famous River Ganga. And we're very, very much looking forward to your talk as well. Go ahead,
Okay,
please.
thank you. Hi everyone, myself Chakresh, working on the freshwater ecosystem, especially on lower stretch of River Ganga, which is commonly known as Hooghly Bhagirathi River. First of all, I would thank all of you to giving us such opportunity to present my work on this platform. Today, I am going to present my work where I look at the freshwater dynamics in one of the major river of the world known as River Ganga. As I show you in the presentation, I will try to explain why this river is lifeline of India and how it is rapidly changing and why will influence the millions of people living on the bank of the river. In this blue line here shows the course of River Ganga from its origin in Gangotri, Uttarakhand. It travels about 2400 kilometers before falling in Bay of Bengal, West Bengal, by traveling through a dense patches of Sundarbans. The river along the way passes through different landscapes where it first travels through the high mountains in the upper coast. Then it enters in the plain, which is known as the Gangetic Delta Plain, which is one of the most fertilized land. And these are the legumes, green legumes and agricultural land. Then it's going through heavily populated cities and industrial cities. It reaches the Varanasi, which is one of the most religious cities in India, which have their own importance. Then it passing many other populated city. It's enter in West Bengal, and through Kolkata, it enter in Bay of Bengal. And but has the river flow through some of the densely populated cities, town, and industrial belt. The beautiful screen that images I showed replaced by the harsh reality of rapid degradation of water, as you seen in these images. Now, the government has several schemes in place to ensure wastewater treatment before releasing into the river or in open area. Indiscriminately, human activity along the river lead the presence of high amount of solid waste in the river. You can say all kinds of garbage material floating around, including plastic, bottle, and other wastage. So why this is a big issue, and what is the real scientific problem behind this? Water from River Ganga are used for all sorts of activity, including irrigation, bathing, and drinking. Additionally, fishing in the water form a major livelihood. Presence of pollutants like I showed you in the previous images before lead to degradation of water quality. To better the water quality, the government come up with the grand scheme which supported the work that I will show up. This scheme is known as Namami Gange, which is started in 2014. And this Namami Gange is attempt to identify the possible sources of pollution in the River Ganga. As you see in this flow chart, there are here sources were classified into two types. One is point source and other is non-point source. And these are the big contributor of pollutants in the river. Point source include the municipal waste and industrial waste, whereas non-point source include runoff from agricultural and rural settlement, open defecation. But has in order to test the point source concept of point source and surface non-point sources, we sampled along the lower stage of River Ganga. The river movement is sluggish, and consequently the impact of the pollutant are stronger as flushing action is less. We sampled along the stretches of 140 kilometers, stopping over the major cities and towns, also in industrial hub. So we identified 10 sites and we sampled several stations at that 10 sites, with including point source and surface water station. In each site, during sampling, we measured the bunch of in situ parameters, with including surface water temperature, pH, dissolved oxygen, total dissolved solid, electrical conductivity, and water flow velocity. And we also collected the samples for dissolved nutrient measurement and the phytoplankton community structure. We then use this information to determine the water quality of each site by calculating the water quality index. Water quality index that can help to provide a numerical value of water. That value, according to that value, we can say whether it is good or not good. And this is what we saw during the sampling. These are the sources of municipal waste, and where the non-point source, where it coming without treatment, the water will become black. And in our finding section, we compared our all measure data. We compared the water quality parameter across the season and found strong variations. As you can see here, monsoon has a higher concentration of dissolved nitrate, which of course could be coming from the industrial runoff chlorophyll concentration is higher in post-monsoon. The water movement in post-monsoon slows down, making the nutrient more available to the biological community. This could help increase the phytoplankton abundance, resulting in higher chlorophyll concentration in the post-monsoon season. Such a observation was also common in other parameters like phosphate, which is high in the monsoon season. This phosphate might coming from the fertilizer added due to the terrestrial runoff during the monsoon, and other parameter also showing there's seasonal variation. Then we did the NMDS plot, which is called non-metric multidimensional scaling ordination plot, showing the 3 clusters based on the measured environmental parameters recorded in point source station and surface water station of the lower stage of River Ganga. A station could be distinguished on basis of pre-monsoon, monsoon, and post-monsoon season. There was a greater overlap between point source and surface water station in monsoon season. The stress value of this NMDS is 0.02. Then we calculated the water quality index. This can be done using several formula and the parameter. We use the pH, nitrate, DO, and electrical conductivity has a proxy of the estimation of water quality. As you can see here, in monsoon, the water quality is very poor as compared to post-monsoon and pre-monsoon. This could be happening due to the coming of very high amount of water from runoff and from agricultural land and other thing, which is change the parameter rapidly change the parameters such as which was used for the calculating the water quality index such as pH, nitrate, DO. We then saw the improvement in water quality in post-monsoon at Dakshineshwar site, but it is only in the one site or one or two station of other site. In again, it is in approx all sites in monsoon water quality is bad than the pre-monsoon and monsoon season. Then I come to our finding. We found one minute, I'm not able to see my screen. Okay. We found a strong seasonal variation in environmental parameters, including DO and dissolved nitric concentration. We also found poor water quality in most of the study sites, which indicate the need to implement a stringent treatment parameter. This mighty River Ganga, coming from the highly populated cities and industrial cities, containing the pollution containing the pollutants due to which the water quality is degraded, fall in the Bay of Bengal in Sundarban, which is the world's largest continuous mangrove vegetation, and it is the UNESCO World Heritage Site and also Ramsar site. Has in the process, river deposited the pollutants and the wastage in this Sundarban, which may degrade the environment of Sundarban. Therefore, it needs to be adopt the robust scientific approach to clean the Ganga river basin and develop long-term ecological monitoring program to access the lower stage of this river. My research is also covered in some articles that from India Today, in scroll.in. Also, it is covered in Department of Science and Technology of Government of India, the Statesman, in the Nature, it's also covered. Thank you.
Thank you, Chakresh, for your insights into your work and into the situation of this river. Are there any questions? Right now, I don't see any. Roxanne, please go ahead.
Yes, hello. Thank you very much for an insightful presentation on one of the world's most iconic rivers. So thank you so much for sharing your work with us. I have a question about how your indice was compiled, because it seems to have a really high focus on nitrate. And is that because of its role in terms of delivery to the coast? Because it didn't seem to include chlorophyll or phosphorus. Am I incorrect there? I just didn't quite understand exactly how it was calculated.
I included the orthophosphate.
What was included in it, please?
Can you repeat the question?
Well, it was just that you had your water quality index and you showed a table with various numbers. And if it was a lower, it was graded as more poor. And if it was higher, it was graded as better, correct?
Yes.
So what I'm seeing here is that the yellow are in better condition.
Yes, yellow in better condition in post-monsoon at one of my site that's as well.
Yeah, okay. So the question is, I'd like to understand how that water quality index was calculated.
We have read the lots of several formula of the water quality index, in which we hello.
Hello?
Hey, hello.
Is there a problem with the connection?
Yeah. We measure the water quality with the help of DO, pH, nitrate, and electrical conductivity.
Okay.
By the
Okay, so what I find quite interesting is, when is it your lowest flow moment? Is it post-monsoon or pre-monsoon?
In pre-monsoon, the flow velocity is lowest.
In the pre-monsoon?
Yeah.
Okay, so there's an interesting, at least where I'm seeing the quality be best, is an interplay of some level of faster flow, correct?
Yeah.
But not sort of a large influx of material from the watershed, right? So there's this sort of interplay of, because your lower flow moment has other types of within system processing going on. So there's an interesting sort of interplay, I think, with your flow moments.
In monsoon, the water quality is better than monsoon. One of the reason is the velocity is slow, and surface runoff is also not coming, which having the nitrate and the other pollutant. That's why in pre-monsoon and post-monsoon, the water quality is little bit better than the monsoon.
Yes, thank you very much.
Thank you. There's another question
you.
from Hans Peter. Please.
Yeah, hi, thank you very much. I wonder, you just showed us chemical measurements. Is somebody in your institute also looking at biodiversity measures? So I'm a microbial ecologist, and I would be interested in whether you also see changes in microbial communities and functions.
Yeah, I am also looking in the microbial community. We are at the Dakshineshwar site where I saw in post monsoon there will little bit good water quality than monsoon, where I checked the microbial community, where I found the Alococera abundance is more than other microbial community. And I think it is the indicator species. It is mostly abundant at that site, I found.
And other biodiversity, do you see a decline when the river is flowing through the urban areas?
And not so in the other area, but Dakshineshwar is one of the famous sites. That's what we catch. We focus on that area. Then I saw that microbial community abundant on particular site.
Okay.
But I forward to continually I am looking on the other urban and suburban area.
Thank you.
Thank you for this question also. There's another question from Abhishek. Do you want to ask your question live maybe, or should I read it? Maybe I give you the opportunity to speak.
Hello.
Hi, yes.
Hi.
Please.
I have one question that in one of the table, there was many sites mentioned and the water quality index was mentioned. So is it in the order from upstream to downstream? And if it is, okay. So if it is, then in one of the sites in the downstream, it was the water quality was regarded as bad. Sorry, it was regarded as good. That is medium to good level. So was it due to any water cleaning effort that has been done?
No,
because
actually,
in upstream it was bad.
We selected the station basis of point source and surface water. So where the people from industry or the municipal waste water is not completely released before treated, and the self purifying purification is not dominant. At that point, the water quality is more bad, and also the municipal waste which is not treated, water is not treated complete at that good level. The water quality is bad, because in one figure I saw at that point water is of the river looking black. So of course at that point water quality is very bad.
Okay, so it depends on the source where you are taking the samples.
Because in the lower stage both side of river heavily population is very high, and they release the municipal and solid waste on the bank of river. So due to this, the water quality is, or some of the station is very low, and in one or two stations where the daily dumping is little bit good, and in proper way the quality is little bit better than that side.
Okay, got it. Thank you.
Are there more questions? I would have a question with respect to maybe more historic situations, because I've heard and read a lot about the Ganga River, and I know that the pollution situation has been very bad more or less always. So compared to, let's say, I don't know, 20 or 40 years ago, is the situation right now worse or better than there?
Actually, on the lower stage of, we are working on the lower stage of River Ganga. Previously, very few work done on this lower stage of River Ganga, and in upper stage where I saw in Varanasi and that side, there also water quality is little bit same about that, but yes, it is slowly degraded. The water quality is degrading. But once the Namami Gange project started, now some changes is coming, and the water quality and the treatment plant is now set up. Then, and it's looking in the it's coming in the picture. It may be in 1 or 2 years the condition of the River Ganga change.
Okay, so there's kind of some hope about the situation improving. That's good to hear.
Because in last 3 years we are seeing at some places they put the net to catch that plastic bottle and other wastage. Also they start the treatment plant which was not working, and in that initiative is already started.
All right, good. There's one last question from Pascal. Hi.
Hello. Thanks a lot. Very nice talk. Very interesting. And I was wondering, did you apply this water quality index, or do you know of other studies who applied the same in other rivers in India, just to get a comparison?
Yes. The same water, this formula is applied in Ganga at the upper stage. They are not applied in lower stage. Till now, in lower stage, there is no study of that water quality. Same water quality index formula is applied in the upper stage. This is the verified by another paper.
And there the quality is better or worse?
In upper stage where they're checking, like Kanpur and in Varanasi, previously it is worse, but now when the project started and the system is changing, there is little bit quality is a little bit good. But as I said, in lower stage of River Ganga, both bank there is very high population is stability, and they are depending on Ganga. That's why this is little bit worse condition.
Okay. And just
okay, Pascal, one more.
Sorry. Okay.
One quick one.
It's I mean, since this quality index is kind of a continuous numerical number, did you think of, I mean, in the end, if you have the landscape features like land use or whatever anthropogenic influence, you could kind of try to figure out what is driving the water quality index specifically, no? Or at least give it a try. You know what I mean?
Okay. Due to anthropogenic activity, water quality is deteriorating, because at some places where there is no anthropogenic activity, that was we selected as the surface water, as compared to point source. The surface water quality is better than point source, where the anthropogenic activity is more high than surface water. So it's yes, of course, due to anthropogenic activity, the water quality is deteriorating.
All right, I'm sorry. I have to stop this conversation or this discussion a bit because we are really running out of time. But you see, there's a lot of interest, and I think it's very good to hear that there's things done now to improve the situation. And from far away, we all wish you good luck for this. And thanks a lot for both of you being here with us today to present your work. Thanks a lot. I hope for the rest of all you enjoyed the talks, and I wanted to share my screen now, but I guess you know all where to register for the next Amplifying Voices session in March. It's going to be the last Thursday in March, and I hope to see you all again there. Bye-bye.